Cell division. In this video, we are going to learn what is cell division, why do we need new cells, what are the different types of cell division, what are its stages, and what are its significances. So let's begin. What is cell division? So this is very simple. The process by which the new cells are formed from the pre-existing cells is called cell division. Now, cell division is one of the most fundamental characteristics of life because cell division is the method which enables life to perpetuate generation after generation. Cell division is equally true in case of the simplest organisms like amoeba, as well as in the highly complex ones such as ourselves. It may be the giant size elephants or a tall coconut tree. Next, what is the need of producing new cells? So new cells are produced mainly for four reasons. It is for growth, for replacement, for repair, and for reproduction. Cell division occur in two ways: mitosis and meiosis. Mitosis. In mitosis, we will learn what is mitosis, why mitosis is called equational division, where does it occur, what are its stages, and what are the significances of mitosis. So what is mitosis? So mitosis is a type of cell division during which a cell divides into two daughter cells, each containing the same and similar number of chromosomes as present in the parent cell. Why mitosis is called as equational division? It is because in mitosis, the number of chromosome remains the same in the parent cell as well as in the daughter cells. Now, where does mitosis occurs? Mitosis occurs in the somatic cells or body cells. Mitosis does not occur in the reproductive or germ cells. Mitosis consists of four stages. Mitosis occurs in two steps: karyokinesis, which is the division of nucleus, and cytokinesis, which is the division of cytoplasm. So, the phases of mitosis are Prophase, Metaphase, Anaphase, and Telophase. Now, during early prophase, the chromatin in the nucleus condenses to form visible chromosomes. Next, the spindle fibers appear between the daughter centrioles. So, this is the cell which undergoes early prophase. This is the cytoplasm. This is the nuclear membrane of the nucleus. These are the chromosomes, and this is the nucleolus. During late prophase, the nuclear membrane and the nucleolus are broken down. The chromosomes are already duplicated as paired chromatids. That is, the chromosome makes a copy to form two sister chromatids. So the sister chromatids get attached with the spindle fibers from their centromeres with the help of kinetochore. So what is centromere? Centromere is the part of the chromosome where the sister chromatids join with each other. And finally, the spindle finishes forming between the poles of the cells. So this is the chromosome. Now these are the sister chromatids. Inside the chromosome, these are the DNA of chromatids. This is the centromere where the two sister chromatids join with each other. And this is the kinetochore fiber which helps the chromosome to attach to the spindle fibers. Review of prophase. So during prophase, the cell looks like this. Now what happens during prophase? The nuclear membrane disintegrates and nucleolus disappears. The chromosomes get condensed, mitotic spindle begins to form, and it gets completed at the end of prophase. The kinetochores begin to mature and attach to the spindle fibers. After prophase, metaphase occurs. During metaphase, the chromosomes gets attached to the spindle by its centromere and moves to the center of the cell. Now, the chromosomes are lined up at the equator. This is the equator of the cell and this is the pole of the cell. These are the centrioles at the two poles. These are the radiating asters and these are the spindle fibers between the centrioles. At the equator of the cell, the chromosomes are lined up and a plate is formed which is called the metaphase plate. A review of metaphase. During metaphase, the cell looks like this. Now what happens during metaphase? The kinetochores attach the chromosome to the mitotic spindle and align them along the metaphase plate at the equator of the cell. Anaphase occurs. So during anaphase, the sister chromatids are pulled apart to the opposite poles of the cells due to the shortening of the spindle fibers. So these are the sister chromatids which are being separated during anaphase. Review of anaphase. So during anaphase, the cell looks like this. Now what happens during anaphase? The centromeres is divided into two. The spindle fibers pulls the sister chromatids to the opposite poles. Now each pole has an identical set of genes. Anaphase, telophase occurs. During telophase, the sister chromatids remain at the opposite poles. The spindle fibers disappear. The nuclear envelope forms around each set of sister chromatids. Nucleolus reappears and the chromosomes reappear as chromatin, and at the end of telophase, cytokinesis occurs. Cytokinesis. So cytokinesis means the division of the cytoplasm. Now division of the cell into two forms identical halves. These are called the daughter cells. In plant cells, cell plate forms at the equator to divide the cell into two, whereas in animal cells, a cleavage furrow forms to split the cell. So these are the cleavage furrow in the animal cells, which is formed during cytokinesis. And these are the cell plates which are formed in plant cells during cytokinesis. Now the daughter cells of mitosis have the same number of chromosomes as the parent cell from where they were formed. The daughter cells are identical to each other but smaller than the parent cell. The daughter cells must grow in size to become mature cells during the G1 phase of interphase. In the identical daughter cells, the chromosome number remains the same, but the cells are smaller than the parent cells. What are the significance of mitosis? So mitosis maintains proper size and shape of the cell. It helps in the growth, repair and replacement of the cells in our body. It helps in healing of wounds. It maintains the same chromosome number in the daughter cells, and it is the mode of reproduction in unicellular organisms such as amoeba and yeast. Mitosis in short. Prophase, pro-metaphase or the late prophase. Metaphase, Anaphase, Telophase, and finally cytokinesis, resulting in two daughter cells with same genetic material.